Welcome
Hello.
to week 2. So to start off with, for 10 minutes, we're going to discuss the post et al. Paper that we assigned for homework last time. So I have some discussion questions here. So first, a really pretty easy one covered in the crash course is what are the key benefits that they identified for cell ag compared to conventional animal agriculture? And people can shout out. Yeah.
One big. I think they identified three, and one of the big ones. So one of the big three was just animal rights,
Mhm.
and yeah, just in general, just much more humane.
Yeah. Anyone else want to shout out some? Easy question.
Another benefit is like public health, because livestock tend to carry a lot of like bacteria and viruses, and also without having animal agriculture, antibiotic resistance might not see as much of that.
Yeah, absolutely. Yeah, or even so there's these common infections like salmonella and E. coli that travel through animals, but then there's also these huge pandemics of infectious diseases that we've seen, like COVID-19 or swine flu, avian flu. A lot of this is due to keeping animals in close quarters and also keeping them in close contact with humans. Yeah, so I think one last one.
Sorry, I just joined. Which two have been said already?
Oh, so we listed animal welfare and public health. So there's one last like big category for a benefit.
Environmental benefit probably. So
Yeah.
like land use, carbon emissions, things like that.
There we go. But this one has a catch. So what is a environmental factor that it's unclear whether cell ag could pose an advantage to conventional meat?
I mean, yeah, slag is pretty energy intensive right now. So you would have to make sure you use like sustainable energy resources if you're manufacturing it at scale.
Exactly. Yeah. Yeah. So that's a really important factor that we still have to optimize for. Awesome. So question 2 I have is relevant today. What are the key considerations that they listed for media requirements? So when we talked about it in the crash course was the cost that media is one of the most the most costly consumable for cultured meat specifically, but it's also a large part of the consumable cost for precision fermentation. You want to find some others?
They also mentioned the BFS, bovine fetal serum, which we talked about too, is currently an animal product and something that we would have to replace for a lot of the sustainability goals we want to hit.
Yeah, absolutely. Or even I realized that peptone, which is commonly found in a lot of yeast broths, it was first started when it was first being sourced. It was sourced from animals, and that wasn't what I didn't even think about how microcultures could use animals, but in recent years there has been a movement towards finding animal-free peptones to basically like digest a certain animal tissue, but instead finding animal-free peptones using yeast extract or soy or wheat. Yeah. Does anyone have some other ones? These are more nitty-gritty.
Yeah, I think another thing was because the fetal bovine serum is so like it just has such a complex matrix of so many things. Like it's convenient at the moment to use that because it works for a lot of different cell and tissue types, but moving forward in the future as we phase that out, you have to think about, and this helps with the cost and scale too. You have to figure out like each individual cell type and tissue types like unique needs, and then you can just like put those things in the serum and find ways to sustainably source those, and then just like get rid of the extraneous stuff, because the FBS has like over 200, I think it said something like that, over 200 different things. You definitely don't need all of those things for any particular cell tissue type. It's just very generalizable.
Yes, absolutely. So if we're moving away from FBS, we need to think about defined mediums, mediums where we know every factor listed that's in that medium, and we know that each of those factors actually have a use in it by the cells. Yeah, that's a really good one. Yeah, so I have a couple more, but maybe people have the answers.
Oh, I was just gonna say also, along with the media, is the fact that they're looking into technology about recycling media, which is cool because I've never heard of that before. So I think that could be a really big part of reducing the cost of cell ag.
Yeah, absolutely. Yeah, we'll cover recycling media a little bit today. And I have a link to a patent for a bioreactor system that uses recycling. Yeah. So the other factors that I have for this question are substrate availability. So there's a difference between these substrates being present in the media and whether, to what extent, those are actually being utilized by the cells. So are they present in a way that the cells can use? And then also, are these components going to be food grade quality? So a lot of the components now are designed to be in compliance with pharmaceutical standards. So those for food are going to be a bit different. And then you even get into if this is going to be used in the final consumer product, is it going to be food safe? Do you need to have allergen testing and then confirm that this is a component that will be allowed to be in a final consumed product? Yeah. So question 3 is about scaffolds, which we're going to talk about for Wednesday. What key properties do you see for scaffolds or other biomaterials used in cultured meat?
Yeah, so this is something that was really interesting to me as I was reading more about it in the article, and I really liked that table they had where it kind of split it up into 3 broad categories. There's sort of like the physical considerations, and then has a lot to do with like shear and stress strains and like the actual architecture and like things like that, like the physical morphology of it. And then you had like chemical considerations, so just how does whatever it is that your scaffold is made of, how does that actually interact on a cellular, maybe like a cellular level with what the cells and tissues you're building on it. And then the third broad category was biological considerations. So things like what happens when you cook it, what happens when people eat it, either raw or cooked, like things like that. And like, how does that interact with actual biological systems? Yeah. It was a really useful table. I was like, wow,
yeah.
this is good stuff.
Maybe we could get some people who haven't spoken yet. So Mringanka, are you here? Or Chelsea, could you give us an example?
I'm here. I'm just struggling to come up with an example right now. Sorry, my Wi-Fi is really bad, so I keep cutting in and out.
Oh, no worries. Yeah, so one of them we've already, there's a couple that we've already talked about as answers actually for the past 2 questions. If you can think of. Ryan? I don't think I can.
Hi, so I'm Yosuke. So actually, I have not. To be honest, I didn't have a time to look at to review the article, but I think definitely cost and also animal free will be needed for scaffold as well, right?
Yeah, absolutely. yes, we want to make sure that that's one of the ones that I had, that we need to make sure that these systems are actually independent of animal sources. And that might not necessarily mean that it's free of gelatin or collagen, because again, we can produce those through precision fermentation, but you would at least have to weigh the cost and benefits of whether you can have that sort of production, and whether it's actually efficient to use those proteins versus perhaps something that's easier to find, such as algae or fungi. Yeah, so the other ones that I had were taste and nutrition, if they're in the final product, and whether it's sustainable. So we want, cultured meat has a quite high bar, because we are trying to have it as an alternative to our conventional animal agriculture. So there's a lot of different aspects that we want it to be better at. So animal welfare is a pretty easy one, but we also want to think about environmental factors, public health, and all of that. Yeah, all right, so last question, which might be more interesting for our business-oriented people. What factors have been important for thinking about consumer acceptance of cultured meat?
Yeah, one that I found interesting, which maybe I guess was a little counterintuitive for me, is it seems like consumers don't want to know too much about the science. So like avoiding saying, I mean, it sounds like lab grown meat itself is a pretty turn off term, but it seems like if you, based on the article, if you explain less of how it's made and more of like either the similarities to meat or the like environmental, animal welfare, any of those benefits, then that makes it more appealing to consumers than telling them about like, you know, the scaffolding or media or extracting cells or anything like that.
Yeah, I think about it, I hadn't thought about it that way, because there's this, so the one of the other factors is like the amount of the more input that you give them, the more accepting they are. So that kind of runs against each other, and I'm guessing how those two factors could be reconciled is less of like the exact information you're giving them, and more framing it as in terms that are comfortable to people. So instead of saying a bioreactor, I've heard people call it a cultivator tank, or thinking about citing things like craft breweries or yogurt as like, oh, these are things that you already know and you're comfortable with, and this is going to be a similar process. But yeah, definitely want to think about framing cultured meat and cellular agriculture and precision fermentation as less technological innovations like Soylent, and more like craft breweries and foods that people already know.
Yeah, that's a good point. You probably want to explain things in a craft way, or like, so I don't know, if you put up like mahogany wood in the labs, then maybe people will be more into it.
Yeah, I don't know if you all saw in the crash course, but there's a beautiful picture of, okay, maybe I might pull it up, of people having a taste test of the cultured meat right outside of the bioreactor facility. There's just like a window, and you can look in and see exactly how your meat is made, sort of like cooking, like when you see the chef over right next to you making your crepe or sushi or what have you, but here you can actually just see the whole entire product being made. Yeah, so I didn't really cite that emphasis to that in the crash course, but I think that's a really cool aspect of people being able to be involved in the entire process.
Yeah, do you know New Age Meats?
Yes.
So I think when they did their first testing, they did it in a brewery because they wanted to show like, oh, our bioreactors are, and I don't think they even use the word bioreactor, but they want to say like our vats are just like the vats that you ferment beer in. And so they had like the whole thing. It looks similar to, I don't know which company this one was, but it looks similar where they had the, I don't know, whatever they're called, like the old like 19th century looking light bulbs. And they had like some copper vats in the background, and it was like, dark wood, like really chic looking. So
Yeah.
that's probably like the way you want to introduce it to consumers.
Yeah, absolutely. Yeah, so this is super meat in their pilot plant. Yeah.
Gotcha.
Sumerganka is saying that for number 4, the role of livestock farming and physical farming or agriculture in the lifestyle and culture of the target demographic plays a big role in accepting cellular ag. Yeah, so an easy way, the way that they generalized this was they found that from conglomerating a bunch of consumer acceptance reports that already been done, they're hypothesizing that Asian countries might see more readily acceptance of cultured meat and precision fermentation compared to America, and America would have more ready acceptance compared to Europe, which I thought was interesting, and I would really love for there to be more talk about exactly what role does livestock farming have in each culture to have this, so that they think that there's this different acceptance rate. Yeah.
Yeah, I also, when we were first starting up the formation of this club, one thing that was a really interesting topic of conversation in regards to culture is that some of these, titles of food that we usually classify our foods as, whether they be vegan or vegetarian, things that are more culturally tied, like halal, kosher, we might start to see the definitions of these things, have to adapt to technology. And already there's been kind of really cool conversations happening. I think, Emily had a really good resource last time.
Yeah, there's already been a few handful of discussions on what would be involved in making cultured meat or precision fermentation halal or kosher. Yeah. So I have already given you all this paper pile link in the folder for project ideas, but I strongly recommend it. It has so many cool papers on cultured meat, from scaffolds and media to religious considerations. It's a pretty cool resource. Chat, yes. Yeah, so the other factors that have been identified that had some saying in some prediction in whether consumers would accept cultured meat or precision fermentation would be if they're young compared to old. Men were shown to have a slightly higher acceptance rate than women, which seemed to be following a general trend that women are more cautious about trying out new foods. So food neophobia is a big factor in whether people would be willing to try cellular agriculture products. And also they found that omnivores were more willing to try out cell ag products than vegetarians or vegans, which makes sense that a lot of vegans and vegetarians have a sort of have developed a sort of general disgust of animal products that is greater than perhaps just the ethical or consideration specifically towards how that meat is made. Yeah. So that's our reading discussion. Thank you for participating.
Can I add one
Yeah.
comment to that discussion? Because I think those are obviously great factors. And when we think about consumer acceptance, we think about the consumer a lot. But there are also some market dynamics at play that go along with any consumer product, namely price.
Yes.
And obviously, the cost, we've been talking about the cost of cell ag a lot and bringing that down so that the price can come down. But I could imagine that if the price is driven so low that it's actually cheaper for consumers to purchase cultivated meat than it is to purchase meat that had to be produced by an animal, especially if sort of the scales start to tip and the economies of scale achieved by factory farms start to dissipate. Hypothetically, that would be a great outcome. Then that could actually really drive huge consumer acceptance, right? Like we consider all these different factors about how you name it, but ultimately consumer decisions sometimes are simple enough as just going for a lower priced product.
Yeah. Or even, I think when I took a class with a food anthropologist, she was concerned that even the opposite that could happen, that cultured meats could become so much cheaper, and people would consider it a lower quality product than conventional meat. So it would be something that like only poor people would eat because they can't afford the more expensive real meat. That is weighing against like, the rich people can eat this more expensive cultured meat that is slaughter free, and so they'll look down on poor people who can only afford slaughtered animal meat. Yeah, so I think that is going to be an interesting development, and I hope that the best happens and we all switch over to cultured meat as much as possible.
And I'm not sure how much you guys already referenced this in the discussion. I joined a bit later, but the geopolitical factors are also very important in this context. So for a country to be able to have its own source of proteins and be able to sustain itself is very important, because reliance on other countries for that type of thing is like a very problematic issue, and can substantially impact trade negotiations and other factors. So if a country feels that the adoption of cultured meat products and alternative proteins as a whole is going to allow them to have food security, it's very possible that you'll just see wide-scale adoption, because that becomes the predominant method of production in certain regions. So, for instance, if you think about the adoption of GMOs in the US or in other nations, a non-insignificant component to that was geopolitical factors, right? And you see kind of the scale of usage of GMOs within the US for common crops is pretty high, to the point it's kind of it's very difficult to find products in general that don't have them, and it has definitely impacted the byproducts of productions and making other things economically viable, and trade negotiations. So
yeah, that's a big one. Yeah, I mean, talking about even specifically cellular agriculture, I think there's talk that Singapore was more readily accepting of cellular agriculture because they rely very heavily on seafood in their diet, I think, and the production capacity for seafood is not keeping up with the demand, and they're a small country, so they're quite eager to have cultured meat seafood. Yeah, or even we had a talk by Paul Mosdiak, who's working for a Piece of Meat in Belgium, and they flew him over from the US to Europe despite the travel ban, but he was considered an essential worker because he is working in cellular agriculture, and cellular agriculture is a form of food production. So that was quite important for Belgium as well. As a small country, they see cellular agriculture as a way to develop food security.
Yeah, as well as like Israel Japan, nations in very arid regions such as Saudi Arabia, Qatar, Kuwait, other like oil nations. Having reliable sources of foods are quite important for them.
Yeah. Okay.
Yeah.
So, yeah.
Oh, I was just gonna say, I did a lot of work on this kind of stuff over the summer too. And it's like these countries are like cell agriculture is like one thing they're exploring, but these countries are looking at like all different types of ways to like grow food domestically. So like vertical agriculture is a really big thing there too. And like Singapore is actually looking at a lot of just expansion of traditional agriculture. Like how do you do aquaculture in like a very limited space? They're trying to like produce a ton of eggs too. So they're trying to figure out how do we get like a lot of chickens in a really small space to just like lay a bunch of eggs. So they're all doing like, they're trying to like reach out to every type of innovation out there.
Absolutely. Yeah. All right. So I will move on for the sake of time. We've got a bit of our discussion, but that was awesome. So now we want to move over to our lecture for the day on media formulation. So first I'll give a little intro to general media considerations, talk a bit about precision fermentation media. Adil is our expert in cultured meat media, and then we'll start working on the final project again, or continue to work on the final project. So what is culture media? From the crash course, remember that it's a solution of nutrients, as well as, I would say, cues that are used to grow and feed cells in vitro. So they have cues to cause cells to proliferate or differentiate or produce proteins, as well as maintain the specific environmental conditions that cells need. So it's made of mainly, at least, it will have a carbon-based energy source like glucose or lactose or whatever is best for that cell, amino acids or a nitrogen source, salts, like including a phosphate source, vitamins as cofactors, as well as, of course, water. So this is our minimal media requirements, or our basal media. And when we think about media, as we said, there's like 3 general purposes that it has. So it has the cues that the cells need to do their specific thing, nutrients to just feed the cells, keep them alive, as well as have an appropriate environment like the pH, the osmolarity, to keep cells happy. So these will change depending on the purpose of that media. So you'll have different media requirements for proliferation compared to differentiation of your cultured meat cells. And you will slightly change the media in precision fermentation depending on whether you're working on product production or you're going to extract that product. And then you're going to have different media considerations based on whether your cells are mammal, insects, seafood, yeast, bacteria, and whether they're muscle or fat or blood, cartilage, feeder cells. So some cool examples that I've seen for how you can change your media is I found another patent by Perfect Day on casein production. And they talk about changing the pH of your media from about neutral pH to 4.6. And that makes your proteins very insoluble. So that knocks out the proteins, specifically casein or alpha lactobumin from the solution. And you can just like scoop off the protein basically from the surface of the tank. And then you can also think about how your media is incorporated into your cells and therefore incorporated into your final consumer product. So when you think about cultured seafood, you should probably incorporate fatty acids, for instance, as those are quite important for the nutritional characteristics of seafood. So ways that you have this formula optimization are first that you do a sort of one factor at a time screen where you either remove or add one factor at a time from your media components. So you might start off with media that gives everything that the cell needs in abundance. So for instance, BSA, and then you start to slowly remove factors and see if that creates an effect. So this will give you initial screening of exactly what components are necessary, at least in some quantity, for your cells to be happy. So then after you have that initial screen of what products you need, what components you need, you have several statistical methods for at what concentration you're going to need each of those components. So the ones that are most generally seen are response surface method, genetic algorithm, and artificial neural networks. And I won't get into too much detail on this because I am not familiar with a lot of statistical methods, but these will give you an example of, these will give you the understanding of how all of these media factors work together to create interactions that you won't just see by looking at by changing one factor at a time. So we talked a little bit in the beginning about media recycling. And this isn't quite a component of media formulation, but it's quite important for still the overall cost of your media, both economically and environmentally. So I have attached to this slide, which of course I'll give you all the slides at the end, which has a patent from Future Meat for their bioreactor system, which includes recycling. So they have a membrane oxygenator that replenishes gases that are used by the cells in some quantities, so oxygen, CO2, nitrogen. And then they also have a dialyzer, which replenishes amino acids, growth factors, and removes metabolites that the cells produce during their metabolic processes, like ammonia, that are toxic to the cells. And we talked earlier last time about how you can do a genetic modification to have the cells themselves actually turn ammonia into an amino acid. But if you don't want that genetic modification, then you can do remove that ammonia with this dialyzer instead. So media recycling is really exciting because it can dramatically reduce your consumption of media per batch. So Future Meat estimates that their media consumption decreased from 2,120 liters to 2 liters per batch, which is crazy. Or in perhaps an easier way to understand, they reduced their consumable cost per kilogram of meat from $21 to $4. So the last general media consideration I want to think about is safety. And a large part of this is just keeping your media sterile before you're culturing, and also keeping it sterile while you're culturing. And there's a couple of ways to do this. First, you want to sterilize your media by irradiation with UV or microwave. And then whenever you are handling the media or the cells or any of the other materials, especially before the culture is really started, you want to use aseptic technique or antibiotics. And it's especially important to keep things clean, I would say, just before culturing, because during the culturing process, most of the process will be automated. It will be done by the bioreactor yourself instead of by manual labor. So there's not as much room for contamination, but also it's important to start off very sterile at the start of your culture, because before your culture has taken over the media, that's the highest opportunity for other microbes to come in and take over that media instead, before there's always say an established culture. And in addition to sterilization, I'll just mention briefly, because we'll talk about this in downstream processing, that you need to make sure that whatever product left, whatever media component left in the final product is food safe, including that it's not an allergen. And you want to, of course, remove any component that is not food safe. So that's our general media consideration. Next, I'll talk a little bit about precision fermentation. So can anyone give me answers for what components we'll need in precision fermentation media? And this will be, for the most part, just the basal media that I mentioned earlier.
Glucose.
Yeah, a carbon source.
A nitrogen source.
There we go, yeah.
Some amino acids.
Uh-huh.
Vitamins?
Mm-hmm. That might be, yeah, that might be needed or not. Vitamins would be, I think, more necessary for cultured meat and mammal cells, and not so important for yeast or bacteria.
Water.
Yeah, water is important. Can you think about like how can you create the environment that the cell likes?
I guess the proper pH. So
Yeah.
whatever acid or base you're using to achieve that.
Yeah, exactly. So yeah, salts or pH adjusters. And there's one that's specific to precision fermentation that you might not think about, but that's going to be these inductors here. And you may or may not need inductors depending on the design of your cell line, but inductors are important if you have, or if you are using an inducible system. So inductors, inducible, where your protein is only produced when its production is activated by the presence of this inducer. So you might want to have an inducible system compared to a constitutively active or always active system for precision fermentation, because when the cell is making all of this protein, it's going to have limited resources to divide. So we found it, scientists have found it useful to actually let cells just divide for a while until they reach their log phase or exponential phase of growth. And then add this inducer when the cells are, when there's enough cells around that you don't really have to worry so much about having a large amount of cells, continuing to produce a large amount of cells, and you just want to focus on protein production. Does that make sense? So thinking about this, like you're balancing your yield per cell with your overall cell vitality to come together and think about total yield, or the amount of protein or product you're able to get out before you're thinking about how the total amount of protein or product that you're able to get out given a certain amount of media or glucose or other biomass that you care about. So given that, these are some examples of media formulations used for precision fermentation, and you'll find that they're found in your average synthetic biology lab. I remember seeing these in my intro to biology lab, 7002 or 7003 for those at MIT. Yes, so there's actually a lot of work already done on precision fermentation in this field that has been around since the 1970s, I would say. So I'm going to turn it over to Adil to talk about cultured meat media. Oh, Adil.
Thanks. Yeah. Sorry. So I think it might be easier if I switch over so I could share my screen. Okay. Okay. Can you all see?
Yep.
Oh.
Yes.
Okay, it looks okay. Great. So I'm going to start discussion on cultured meat media and in general just media formulations for animal cell lines. So to kind of first say why is it important to really talk about cultured media? Media costs are estimated to be somewhere between 55 to 95% of the cost of cultured meat, and the estimates have changed over time as the cost of media formulations have gone down. But so this is a little bit of an old estimate, but it is one of the most important cost drivers. So developing low-cost scalable media formulations is one of the main challenges in the field. And our capabilities to really executing this well is going to define not only the price and accessibility of meat, but also the quality of the meat that will be made. So what I want to do is really kind of take you through a bit of the history of animal cell culture, because I think while learning the history, you understand the main strategies that people have used, as well as the key discoveries that led us to the media formulations that we currently have today, and kind of a roadmap of how we might be able to go on in the future developing new media formulations. Okay. So one of the first main strategies that was that people tried is kind of a reductive approach, which is really about trying to define a solution that works well and then reducing it down to its simple, most necessary and sufficient components to have a solution to your problem. And the way this kind of started was back in 1882, people were interested in growing cells, and they realized if you had basically a mixture of balanced salts that mimics the physiochemical ionic conditions and concentrations of cells, that you can sustain cells in vitro. And the first group of people to do this developed Ringer solution and Lock solution, and there's a couple of other main ones. But towards the end of that period, people discovered that if you used lymph material from lymph nodes, that you can also grow cells using this. And it was actually sustained a bit longer, so that there might be something specific about the biofluids of animals that allows for cells to grow longer, which led to people trying out actually using blood and different fractions of blood to grow cells. And this was substantially more successful. Now instead of getting a couple of days of cells being sustained in vitro, you were getting a couple of months. And that was really how we kind of started to signify the start of animal cell culturing. And although it's debated, the first person to do this was Margaret Reed in 1908. So I think that's really interesting and less highlighted discovery of female scientists. And from there, the people started to actually identify components in serum that would sustain the growth. So what was it in the blood and the fractions of bloods that were important and valuable that made the cells live longer? And some notable conclusions that were drawn was that modulating the glucose concentration had an effect on the cell growth and viability, as well as specifically the protein fraction within the blood was one of the most important components. And so now you see kind of the reductive approach at work, where people had a working solution and then tried to see what are the necessary components. Okay, so basically what during this period, what people were identifying is called natural media solutions today. And it's basically plasma, serum, lymph, and any types of fluid or tissue extracts from living organisms that can be used to grow cells. Okay, and this is basically kind of all of the other components that are important to the cell viability and vitality. Usually the ones that are still used today are serum, and that's a really important point, that a lot of the stuff that was done, all the way back in the early 1900s is still really important to how we think about culture media today and how we use it today. And just a refresher on exactly what serum is. Serum is basically if you centrifuge out blood and you kind of take out all the red blood cells, white blood cells, platelets, and anything that's associated with clotting, and you have the remaining components, that's serum. So it's a little bit different than plasma, but quite similar. It doesn't have some of the other clotting factors. And in formulations today, you'll see somewhere between 5 to 20% of media being composed of serum, and the rest being composed of the basal media. And this is important to notice because the main serum that's used is called FBS, and the method of production of FBS is really, it stands for fetal bovine serum, and it's made by extracting blood from mature fetuses during the process of slaughtering cows. And about 8% of cows are pregnant at this time period, and that blood extract is used to create the serum. And because of that, there's generally a fixed supply of FBS, a formulation that you can have at any given time. And also there's a bunch of other problematic issues associated with this. And I went over this in the crash course, but I just wanted to highlight them again, is that FBS is not animal free. The process of getting it is, really ethically questionable. It's because of the fixed supply, it's very expensive, it's right around $1,000 per liter. And just like any other natural media formulation, because it's being harvested from an organism, there's batch to batch variability, and it's not chemically defined, so there's reproducibility issues. Okay, so that's kind of the sphere of all the natural media formulations, and it's has history all the way back in early 1900s. And we see a really big shift in how animal cell culture is done, kind of in the middle of the 1900s, when people start to develop the first sets of immortalized cell lines. Because before then, there wasn't outside of some research considerations, there wasn't a ton of uses of animal cell lines being grown, because there just wasn't a way to have the same cell line being used over and over again. So studies, reproducibilities, there's a lot of issues around it. So once people figured out how to use carcinogens to develop immortalized cancer cell lines, the demand for media formulations and the need for new media formulations became much larger. And notable ones you guys probably might know of is like HeLa or mouse L cell lines that are still in use today. So what basically happened after that was people continued on using this reductive approach. They tried to basically say dialyze serum. So dialysis is basically just a process of separating and filtering out serum to get smaller and smaller in quantities of the serum components and make larger and larger components of the basal media. So you could have more and more basal media and understand exactly what are the components in the serum that are necessary, because the basal media is all synthetic, and the serum is the component that's actually, again, got from a biological material. So if you could reduce the serum by filtering it and having smaller portions, then you could have larger and larger portions of media formulations. So again, this reductive approach at work. And at this time, a second strategy started to be employed as well, which is kind of this synthetic approach. So if you think of kind of the reductive approach as a bit of like a top down, where you have some working solution, you try to work down to find the base components. The synthetic solution is more of a bottom up, where you try to use your knowledge of the space of possible solutions to build up something completely synthetically that can be used as a media that works well. And the thing is, though, to get to the point where you really do synthetic approaches, most of the time you need a bit of reduction that works, because you need to understand what is like the space of possible solutions. It's really hard to develop a synthetic approach without knowing what are the components that might be needed for. And so you see this kind of split where people really work very diligently on synthetic approaches at the same time at the same time point of 1946, and people are really trying to develop protein-free media formulations. So where instead of using full-on proteins that are coming from some type of organic source, you're saying let's use like small peptide chains or amino acids. And because remember, one of the most important components of serum that was being used was the protein fraction. And a bit later, people started working on fully chemically defined serum-free media formulations. We're saying, okay, now that we have a good understanding of exactly what are the amino acid components and other components that are being added, can we make it completely in a chemically defined way? So we know exactly every single component that is added into your solution. So that kind of can be summarized into the second section of synthetic media. So synthetic medias are basically media formulations that have that are mostly being produced by your knowledge of the exact components and adding them together. So you could say it also includes serum-containing media, where you have mostly basal media and then a bit of serum being added. Then there's other types of media formulations, which are serum-free media formulations, where you take the protein fraction out of serum and then use that just with your basal media. Xeno-free media formulation is not really important right now, but it's basically similar to serum-free, except you're using just human components instead of from an animal source, mostly for therapeutic applications. And then the protein-free and the chemically defined, which we talked about, the protein-free basically is referring to using hydrolysates, which are basically just short peptide chains and amino acids that you can extract from a variety of different sources. Basically, anything that has basically any biological source, and that you can get it from yeast or plants or coli, wherever. And then chemically defined, which is saying now using recombinant proteins and having very exact knowledge of the concentrations of everything inside of the media. Okay, so that leads us into now more of the modern age, where you have cultured media production, specifically for animal cell lines that are being used to make recombinant protein. So right around this time period, people were starting to use cell lines, or even either using E. coli to make therapeutics like insulin. Okay, but the difficulties of using E. coli or yeast to produce recombinant proteins is that the E. coli can't actually add some important modifications to a protein for the proteins to work well. So they call these like post-translational modifications. Specifically, the one we're referring to is like glycosylation, which is adding sugars onto your protein so that it functions properly. And animal cells do this a lot of the times, and it can have a dramatic impact on how effective your protein is. And so once people started to realize that not all the proteins that we want can be made from E. coli or yeast, and we might need to use animal cells, that then there was a lot of work being developed on actually making high quality media formulations for animal cell lines. And that continues on to this day. And then you see basically another hallmark, which is people really trying to develop new media formulations for stem cells. And this is important because stem cells are really a lot of the cell lines that are actually being used for culture meat production. Okay. So for this specific component of like animal cell lines being used, some of the industrial advances that were happening where people were using a very specific cell line. The most common cell line that's really used is CHO cells, which are stands for Chinese hamster ovary cell lines. So a bit strange, but that's the one they found. One like was really good at expressing proteins, and we had a really good sense of all of the viruses and the safety considerations that were important to the cell line. And basically during the time period of 1986 to 2004, you saw a 10x in the amount of proteins that like the single cell, like CHO cells, were able to produce. And basically a lot of the ways that was really done was really some of the techniques that Emily mentioned, which is really analyzing all the different components that were involved in the media, and trying to understand the landscape of, what mixtures of different concentrations of these components would yield the best production. And one of the main things that CHO cells are used to produce is antibodies. So you can see in this example, just two components that are being used or looked at to maximize antibody production. But there are, as we listed already, there's much more than just two components that are involved. Okay, so again, to now talk about a bit of the stem cell serum-free media formulations, there was a set of different ones that were produced, and I just wanted to list them out. If you guys want to, look and read into detail about each one of them. But there's various changes on different concentrations of some of the important factors, which I'm going to start listing out now. So this is kind of the playbook if you want to take out serum for your from your media formulation. Okay, what are the components that you're mixing and matching, and like to make your different media formulations, to do your optimization, you have to know what are the factors kind of at play that you can use to really substitute well. And so this is a list of some of them. Hydrolysates, which I mentioned, which are important for the protein-free fraction. They can come from a variety of sources, as I mentioned. They are really low cost, but you can get a lot of variability in them, depending on how it's sourced and how you do processing. Then there's growth factors, which are very important for proliferation, differentiation, secretion, a bunch of different things. And the worry here that you have to consider is about where you're getting your growth factors, whether it's recombinant or not, because if they're recombinant, it can be very expensive. And if it's not recombinant and it's coming from some animal source, that's also a consideration. You have to consider specifically about contamination issues or ethical issues from your sourcing and reliability. And then there's hormones. So one of the main ones to mention is insulin. That's really important to enhancing proliferation of your cells. And the stability and cofactors needed for it to work effectively are things that you have to balance. Okay, and then the next set of really important ones are some carrier proteins. So these are things like albumin, transferrin, lactoferrin, and what they help is bring the any lipids, trace elements, vitamins, or other components that are needed for your cells to work in a desirable fashion. And this is all really cell line specific and about what is needed and what concentration. But there's also pros and cons you have to consider here, where they're good at, potentially neutralizing toxins by binding them well and balancing out any reactive oxygen species. But just as their ability to carry in useful things, they can carry in contaminants. So you have to make sure you know where you're getting them from.
I have just a quick question
Yeah.
about this, if you're taking questions right now.
Sure. Yeah.
Yeah. So just for clarification, when you say that the lipids, commercial lipids, tend to be animal derived, do you mean that it's like a byproduct of the livestock industry or something else?
Yeah. No. That's generally where it comes from. Yeah.
Gotcha. And is there any work being done to try and change that? Because I feel like that would have to happen at some point.
For sure, yeah. And so lipid production is, a bit challenging because, lipids don't work well with water, right? There's a lot of separation, and depending on the density, can float to the top of your production system. So lipid production, there's a couple of main ways that people are really looking to it. One is algae based. Algae are really good at producing very specific types of lipids. Then there's other specific types of microbes that work well with production of lipids, and one of the main ones that people are looking into now are like palm oil and other productions of problematic fat-based ingredients that are used throughout applications like food productions, and then there's more like animal cell line production methods. So you're talking about now saying, let's have an adipose cell, so like fat containing cell, and producing that in the same fashion that we kind of discussed. And lipids are really important for cultured meat production, because the taste and flavor of meat really comes from this. But yeah, a large part of the way that lipids are used, at least in the media formulation component, is animal derived. And lipids are really important because the membranes of your cells and a lot of how the cells interact is dependent on the lipids that are in there. So it's an important factor in consideration. And there's a variety of approaches that are taken. And plants as well make lipids, so they're sourcing from that. And then the another one is the vitamins and trace metals. Trace metals is something people really didn't understand or figure out was essential until they realized it was actually through methods of like contamination. Like trace metals were in the water sources that people were using to make media formulations. They realized once they changed or made it in different places that it didn't work the same. And the importance of trace metals became apparent, and really they are important for cofactors. And this is the same reason why you need to make sure you're getting like micronutrients in your diet. But understanding the quantities and the solubility for these components is a bit challenging. And then there's a couple more factors that I'm just, for brevity, I'm not going to go into detail about. There's polyamines, reductants, protective agents and detergents, and adherent factors. Depending on the cell lines and the use case of how you're producing things, these will be important. Okay, and so really when people are trying to develop new media formulations, there's a sense of, for each one of those components I mentioned, what are the range of like valid concentrations that you could have. And then it's about understanding what's a good starting point for these quantities for the cell lines that you are interested in, which is based off of historical reference of what media formulations were used, and some understanding of the metabolism and a bit of like, educated guessing that happens. And then there's an optimization that happens where people say, okay, let's actually explore this combinatorial space to identify what is the optimal media formulation for the specific task of interest. And so this one here is still showing antibody production, but if we're talking about cell lines for clean meat industry, you're likely talking about proliferation rates and capabilities for differentiation, and also some more subjective things about taste and flavor, other things, which you might be wanting to optimize for. And the process for this, this is just kind of a high-level overview where you have formulations of your raw materials that goes into your how you actually then decide what your combinatorial space is that you're exploring, which is the DOE. And Emily mentioned some of the strategies. You one factor at a time analysis, or you do like combinatorial analysis of the space. Then you have high throughput methods that people will use to test out all these different combinations. Then you do kind of a factor analysis of what is the most important things. Then there's approaches where people will look at what is the media that is left afterwards from your cells, and you understand what is the metabolic, understanding what the metabolism of the cell was. So what was it using? What was it not using? At what rates was it using it? They call that like spent media because you've used it, it's spent, and you can see what's left. And then you kind of fit your media for the purpose of interest. Okay.
I had another. Sorry, I had a quick question about that slide too, if there's time.
Sure.
It looks like as the state of the industry right now is at somewhere along the line, it's clearly a lot of opportunities for things like machine learning and combinatorial statistics to come in, but somewhere along the line, they're actually like testing it out in real life. But are there any developments in terms of doing this purely computationally, like just using models? Like, do we know enough about how things work, I guess, to just do it that way?
That's a great question. I would say no, not at this current time. Do we have enough for you to kind of get to the point where it's completely in simulation? Simulation driven exploration is definitely an approach that can be taken, where you understand a bit about what people tend to refer to as like the metabolic flux. So how different components are moving in and out of your cell to get a sense of the rates of components inside of your cell and how it will grow and divide. But we don't know enough about cells for us to do this completely in silico, as well as a lot of the cell lines that we're looking into have quite a bit of variability because they are primary cell lines from specific organisms. There's biopsies that are taken into consideration. So these really make will complicate it if you're trying to completely do in silico.
Gotcha. Thanks.
Yeah. Okay, and so now, so all of that I kind of mentioned before, and this one is really more of a synthetic approach. Remember, I kind of talk about the two strategies. This one's like also very more of a synthetic approach. Then there's more of a reductive approach that people are also taking, which is saying, okay, so this is a stem cell serum-free media formulation that is currently working and being used, and it has these couple of components. This one specifically is called Essential 8. Okay, so let's just try to figure out what are the expensive components, reduce the cost of them, and just have cheaper media formulation that we could scale. And this type of analysis, identified that specifically there's like two growth factors that are very expensive and account for like 96% of the costs. And so if we could reduce these, then we should be in good shape. And so the proposal here was saying, okay, one, can we engineer our cell lines or adapt them to not need as much of this media formulation, or specifically of those growth factors? Then can we make these growth factors in a different way, specifically trying to make them using E. coli instead of mammalian cells? So one of the biggest reasons for their cost is that specifically the FGF2 and TGF beta is that they were being made using mammalian cells. And that is very expensive versus, for example, insulin, which is made using E. coli and other bacterial systems, that it's much cheaper per gram. And so similarly, can we make these growth factors in a cheaper host organism? And then if we combine these two strategies, likely we should be able to see a reduced cost to about like $5 per gram. And so this was the start of where people actually were doing exploration. And this is an example of a one factor at a time analysis that was done on human-induced pluripotent stem cell media, where you can kind of see, this is the list of components that I mentioned. You guys can see my mouse, right? So list of components down here, and they're saying, okay, so let's look at each one of them at a time, change the concentration, and then see what is my, what does my growth look like, to understand what's needed, what's not needed. This analysis, for instance, is interesting to see. They say, okay, if you have no insulin, your cells die. So you need to have insulin in a very specific amount. If you don't have, this other growth factor, your cells kind of don't do so well. But some of these other ones, you get somewhat growth even when it's not at the amount that we've set. And this one at a time analysis is helped to use to say, okay, what is the amount of each of the media formula, each of the components that is necessary. It misses out on some of the combinatorial effects, so how the different concentrations of each of these components interact, but this method of one factor at a time really simplifies the analysis. Okay, so I mentioned this one because this group also did used E. coli to produce FGF2, then TGF beta, and other growth factors that would be necessary for the cells. And they showed that they could make a media formulation that was, quite simple and reduced in the concentration, and using this at scale to be right around $11 per liter, and still maintain, pluripotency after 100 passages of the cells, which is a really great accomplishment, and one of the few more like academic publications of exact media formulation. One of the difficulties in this field, if you're really interested in looking more into it, is that for media formulations, people start calling them not even chemically defined, even if it is chemically defined, because commercial companies who make these media formulations do not post the ingredients. Because if you post the ingredients, then other people could just make them in their own lab, right? And a lot of media formulations have very large markups, talking like more than 95% margins on media formulations. So for the sellers, it's a good industry to, a good product to sell, but for consumers, it's very problematic. And so this is great because it's a academic publication showing the exact formulation here. Okay. And so what you see is that this it was showing these are kind of the currently used serum-free media formulations for stem cells, which hover around $400 to $600 per liter. And through the strategies that they showed, you got a huge reduction from using E. coli, and then another reduction from doing it at scale and reducing the quantities of factors, which is similar to the technical analysis I showed, where you saw reduction. It wasn't as significant as this like $5 per liter amount, but still quite in symmetry. And then the other main techniques for reducing media formulation that at least this study suggests is saying, can we drive down even more the cost of all of our growth factors by changing the organism that's used to make it? Can we, instead of using medical grade ingredients, can we use food grade ingredients, especially for the basal media where you have like ascorbic acid? Can substitute from something else? Is okay, can we just use something from oranges or whatever, right? Or if we wanted to look at NaCl that's medical grade, and said, can we just use table salt and reduce our costs a lot? And that's what you see in strategy E. And then, similarly, so the ascorbic acid, and then switching to cheaper buffers to get a very, very low cost, talking about fractions of a dollar per liter, which is where you really want to get to clean meat to be super like economically viable. And then there's kind of more
quick question if there's time.
So wait till the end and we'll
okay, sounds good.
Yeah. Okay. And so, because I'm almost done. So I just also wanted to highlight a couple more exotic strategies that are really interesting to think about, because I kind of been more focusing on this, reductive approach or synthetic approach. And then there's some people kind of coming in a little bit left field and saying, hey, can we make a system that looks kind of like this? Can we just try to mimic what is done in the human body and basically have different containers for each of the cell lines that for different organs, and organs are working together to basically create a system that allows for what can be considered as immediate formulations for your cells to grow. And what that looks like is basically you have different combinations of these organs producing different factors that is used to grow your cells, and it changes at different times. So you have proliferation-induced combinations, differentiation-induced combinations, and then organization-induced combinations for you to be able to get some type of structured meat product. And this work is being done, or this system is called CullNet, because it's like a network for cultivation, and it's being done by what is it again? Interaculture, I think, is focusing on this as a Japanese company. Okay, and I've mentioned a lot of things, but there's still a lot more other considerations that we really just, for time considerations didn't get into, such as, okay, if you want to actually scale up your media formulations, it's not a linear formulation that will work, right? If something that works on the bench, that works at small scale, doesn't necessarily work on large scale in a reactor, and you need to tune it to your system. Also, the type of reactor it's used substantially affects the media formulation that is actually being made. The sourcing of your components and like where you're getting your feedstock and the volume and the pricing sensitivity to these components are really important factors to consider when you're producing at bulk. And then waste management. So the spent media that's being produced from these cells can have some cytotoxic factors or other components that will actually affect people, because now you're talking about cell lines that people interact with. It's not just like bacterial components. There's some sensitivity now to it being an animal product that it could actually affect and translate to human. We talk about diseases. And I think the last topic I wanted to get into a bit was some of the commercial considerations. There is a variety of different companies that are out there that are producing beef, chicken, seafood, pet food, pork. Others, these are more exotic ones, like kangaroo foods, and each of them need their own media formulation. And it's kind of growing to be its own strata within the like B2B market of selling things. And there's a couple of key players here that you guys, if you want to, look into and see how they're approaching the problem for making media formulation to help all of these different players develop a media. Because especially if you're just starting out, having a media formulation that will work and get you going is a non-trivial problem, because if you are like trying to make a shrimp cell line, you're not going to really find many publications on shrimp cell lines. If you're making, I don't know, if you're trying to make oysters, like there's not, right? There's not going to be too many published papers on this topic, and people helping to actually develop these novel media formulations is really important. And there's some interesting pros and cons to working in that space, specifically. Cell lines all kind of have optimal variants. If you're trying to sell media formulations, that could be good. Pricing and cell densities and quality are really important to these companies. So having high quality media is important. And there's a lot of other markets where media formulation is also important that we didn't really talk about, but stem cell therapies is a very, very large potential market that's growing, and IVF treatments where you need to grow cells, and they also want, high quality optimal media formulations. But there's quite a bit of cons if you're trying to actually make this on commercial scale, which is the market for clean meat. Specifically, the media formulations is quite small. There's a lot of competition, as I said, from different startups that are already working on this, and big companies like Merck, and I think Thermo Fisher might be working on some clean meat media formulation. And most of these companies kind of want to do their media formulation in-house. But it's an interesting space, and I think media formulation is one of the most important factors for clean meat. Okay, so I think that was it.
Yeah. So my question from before, as I was thinking about it in the last few seconds, I found a different way to. Oh, sorry. Is there not time to?
Yeah. So it is 2:26, and we can answer the question. I would say right after, but first, Adele, can you move to the next slide?
Yeah.
Or it's 2 slides. Sorry, we ran out of time today. So I want to talk about the final project timeline. So last time you started researching the background. Today, I wanted to get into making a working abstract, but I'll leave that for homework. That you have a working abstract and start planning or ordering your materials. So that by next week you can start doing your hands-on experiment if that's what is part of your project, or you start writing your project final proposal. And then remember, week 4, we're presenting our final projects. So then next slide, Adil.
Yeah.
So next time, be prepared to share your working abstract and a little bit about your topic and project stage. And be sure to start planning, ordering your materials. Here are 2 sites that would be quite useful if you're doing any hands-on project for fermentation or what have you. Okay, now we can ask questions.
Thanks. Sorry. Yeah, my question was I thought of a simpler way to phrase it, and it's really comes down to, we keep hearing the same things when it comes to the production of the pricey components in cell media, like usually it's like yeast, E. coli, etc. These are like the main prokaryotic organisms that are used to sort of ferment it. And I'm just wondering, is there an active space right now looking for new, maybe exotic forms of prokaryotic cells that maybe one day it'll be like a miracle, like, oh, this one can actually produce it even more things, or it's like even more efficient or something like that?
Yeah, so that's a good question. Okay, so in terms of productions of the different growth factors, because if we're talking about the main expensive components in media formulations right now that can be produced in different methods, it's growth factors.
Gotcha.
And there are a variety of different ways that people are exploring production right now. Some are using algae systems, others are using E. coli and yeast. And even when you start using E. coli and yeast, you're talking about substantial reductions in price to the point where it's really competitive and you don't need to make a bunch of other changes, but people are still looking into, because there's still reliability issues that you have in those instances. And so people have developed different plants that can actually make recombinant proteins. A recombinant, like plants being used to make recombinant proteins is not new. It's been used for different therapeutics in the past. Specifically, lines of tobacco is one of the most effective that's done at scale, and people are using that. There's a couple of companies specifically out in most of this work for media formulations happening in Europe, and some of the key ones are in France, and I can send them to you, but they are mostly focusing on plants to do this. And the key thing is actually what's interesting is that growth factors, they're really can be used for a lot of different verticals. It's not just clean meat. We talk about like stem cell therapies. This is the main thing. A lot of the companies are trying to in tangent work on growth factors for clean meat, and then work on it for stem cells. But gosh,
are there any differing, like, are there any extra considerations when you make the growth factors for clean meat as opposed to the stem cell therapies, like something to do with nutrition or anything like that? Or are the growth factors, like, I guess, small and fundamental enough that it doesn't really matter?
Yeah, so I mean, the growth factors are usually just it's like a protein. So you know the exact sequence of what it is. So the main consideration now is, depending on what you're using to produce it, it's the quality of purification you can get it at, and the yields, which dictate kind of the price that is needed for it. Another one will be more important when you talk about clean meat is the scale that you can produce it at, as well as in the proximity towards production facilities. Because if you're producing very, very large bulk quantities, having it near in-house or next to in-house is important. And this is kind of generally for feedstocks as a whole, kind of having them closer to the production facilities is really important. And the other is kind of, so downstream processing was what I meant when I was talking about purifications, and because you can have a much less pure mixture if you're making clean meat. For example, if you have a plant that has this growth factor, you can just like chew up this plant material, then put it in your media formulation. You cannot do that for therapeutic. That's not a viable thing, but it can make it so much more cheaper, especially. So the main cost component of making recombinant proteins for like yeast or E. coli, some bacterial system, is not really how efficient the organism is at producing it, but the infrastructure costs, right?
I see.
Because there's a fixed cost of having a reactor, having it cleaned, having all the labor and energy and water usage that's associated with it, that is going to fundamentally limit the price point that you can get at for a product. Which is why most of the times, if you're talking about recombinant proteins being produced, it's usually for therapeutic. Now that you see like clean meat and like precision fermentation products being developed, you kind of have to drive down the cost, but there's only so low you can get that price point before
I see. Yeah. It's kind of a limited area. So the potential, it sounds like right now, given what we have, is relatively limited then in terms of driving down that cost.
Yes.
Because of like the fixed infrastructure, fixed energy costs and stuff. Yeah.
Yeah. So now the question is, is there potentially different organisms that allows you to use a different, cheaper infrastructure? Okay. Because now if you can break into that, you're saying it's a different kind of ball game, because now that fixed cost becomes a variable cost, right? So you can imagine, for instance, trying to use algal systems that can work in very, very cheap containers, or there's open sea facility, or like open lake facilities that are being used mostly for biofuel productions. But like, potentially you could try to see, can you have an organism that allows you to use cheaper and cheaper infrastructure to drive down that cost as low as possible? Yeah.
Cool. That's actually really helpful. Thank you. Okay. Yeah. Interesting. That's cool about the plants. I'll look more into the if you actually, I can send you an email later on if you had any sort of resources on European firms that are doing stuff with the plants, because that sounds cool.
Yeah. Yeah.
Thank you.
Yeah, anyone who needs to, you're welcome to go. We're just here if you have any questions on the content covered or the final project.
Yeah, I just had a question on the project. Yeah, so I'm an MBA student and not doing anything super scientific.
Yeah.
So I was just gonna like make sauerkraut basically.
Sounds good.
Which I saw that was an option. So like, I mean, what does that mean in terms of like making an abstract and like all the other phases of this project if we're just like, you know, making sauerkraut?
Yeah, so I would say instead of a abstract, although you could, if you wanted to, you could do things like try some experiments with your sauerkraut. So try out what happens if you include different vegetables or different salt concentrations. Try to get some science in there. But what you could just do instead is give us a little progress report of like what stage you're at in making your sauerkraut. What have you learned about the science of sauerkraut so far, or any other aspect of the sauerkraut that you find interesting? Maybe like different recipes for sauerkraut that have been made by different cultures, or yeah, different types of fermentation for vegetables. Yeah, and also just start planning ordering your materials for that.
Yeah, sounds good. No, my jar is fermenting already, so I think probably great. Phase 1 is just going to be like making sure it's food safe. So
yeah,
like, I guess my first round will probably. Sorry.
Sauerkraut is pretty. I shouldn't be too worried.
Oh no! So I tried this last year, and I had like mold growing on my sauerkraut. Oh. So I definitely did something wrong. So this try, I guess my first attempt now is making sure that does not happen. That was probably just me screwing up, doing something wrong. And then I guess maybe after that one, then I'll try to do some of the more scientific experimentations.
Yeah, I will say. You have to keep things really sterile. And as I was talking about earlier with safety, it's important to start off sterile, because when you are, so like being sterile as in your jar is sterile, you've washed your hands, if you're trying to mix the sauerkraut with your hands, because cultures might get on there that are not the cultures that you would like to have on there if you're starting off with an empty slate, and you don't have, so like as sauerkraut progresses, it becomes a method of preservation, because there's basically bacteria already in place that will prevent any other different microbes that you don't want from taking over those vegetables. But at the start of the fermentation, that's when you can have just this plate of yummy food from bacteria or other microbes that can come in and start eating that. So definitely start off very clean. Figure out there are different ways that you can find online for how to sterilize your jars. But yeah.
For sure. I tried boiling all my stuff, so that does it. But yeah.
I also wanted to mention that you might also find that this paper specifically is a great analysis of. I only touched I only talked about like less than half of what this paper goes over on a techno-economic analysis of media formulations and the viability of cultured meat as a whole. And I think this is a space maybe as an MBA student that you might find really interesting. And I don't know, I guess you already have a project, but that you could look into it.
Or the GFI's paper on precision fermentation that I just put in the chat is also like specifically, they talk about precision fermentation as also relating to other types of fermentation, including sauerkraut. Yeah, so that might be interesting more as a market business aspect.